How's everyone who's watching this? This is my 32nd video on the channel, and today we're going to be beginning Chapter 2 of Intensive Science and Virtual Philosophy by Manuel DeLanda, the actualization of the virtual in space. Last time, we looked at the role of vector fields, modality the virtual, and state space ontology in Deleuze's work. In this video, we'll begin to unravel the mathematical metaphors DeLanda used throughout the last chapter, and see what it means for multiplicities to be actualized. If you haven't seen the other videos in the series and don't have at least a basic understanding of Deleuze's ontology, I really recommend going back and watching them. With that out of the way, let's get into it. DeLanda begins with essentially a recap of what we discussed in the first episode of the series, where he used the metaphor of a topological space being specified, transformed, and progressively acquiring detail until eventually giving rise to the metric Euclidean space we occupy, in order to show how the virtual is actualized. There, in Chapter 1, The Mathematics of the Virtual, DeLanda began to unravel the metaphorical side of this notion by introducing the idea of intensive properties, like temperature and pressure, being the things that undergo differentiation to give rise to extensive forms. However, there he only introduced the idea. It's in this chapter that he wants to develop it. In terms of what we're actually going to be talking about today, he says,
in the first part of this chapter, I will discuss two different aspects of the intensive, each illustrated with a specific individuation process.
These two aspects are what he calls population and rate of change, which he opposes to the essentialist concepts of fixed types and ideal norms. To explore them, he invokes the myriad of ways biologists explain the individuation of living organisms into species as a case study. For much of at least Western history, species were seen as perfect examples of so-called natural kinds, to which there were two main approaches: that of essences, exemplified by Plato, and that of natural states, represented by Aristotle. In the first case, organisms in a given species are seen as representing, albeit in a flawed manner, some sort of perfect idealized form. For example, all house cats can be grouped together because they are representations of a transcendent, the most cat-like cat that could ever possibly be a cat. Concerning the second case, it's simply a way of classifying things together based on similarities in what they do. A cat is a cat because it tends to do certain things. In both, the main idea is that there is some sort of abstract exemplar that things are supposed to be like. However, in DeLanda's words,
Charles Darwin broke with this tradition by showing that species, far from being eternal archetypes, are born at a particular historical time and die through extinction in an equally historical way.
what he wants to bring our attention to is the somewhat controversial and quite recent idea that species are individuals in their own right, a notion that takes Darwin's breakthrough and runs with it. The main voice behind this view is a biologist named Michael Gisselen, who argues that species don't represent higher ontological categories when compared to their component organisms. In other words, species aren't like essences for what make them up to exemplify. Instead, they're just like bodies that take individual plants and animals as their cells. The other important feature of Gisselin's species individuals is that they only exist through the causal relationships between parts that make them up. As an example, a new species is said to form when two populations can no longer reproduce with one another. As DeLanda says,
this reproductive isolation is a causal relation between the members of two subpopulations, and, moreover, it is a relation which must be maintained throughout time.
This isn't to say that organisms and species are the same, however. In terms of both space and time, the latter obviously have a much larger extension than the former, more area covered, a longer lifespan, etc. Rather, is to propose what's known as a flat ontology, a way of viewing being that gets rid of hierarchies, like the one usually applied to organisms, species, and genera. DeLanda describes such an ontology as being exclusively made up of unique, singular individuals that do differ in spatio-temporal scale, but not in ontological status. However, it leaves us with something of a conundrum. How exactly does the whole represented by species come to be? The answer is intensive processes, defined as such because of their heterogeneity and their population-based description, something DeLanda hopes to elaborate on through his exploration of so-called population thinking. This way of thinking is described best by one of the most influential figures in the synthesis of evolution and genetics, Ernst Mayr. Comparing it to typological models, like those based on Aristotle, concerned first and foremost with categories, he says that populationism stresses the uniqueness of everything in the world, only grouping things together based on statistical variations. The typologist, in Mayer's eyes, sees only a limited number of fixed, unchangeable archetypes that underlie variability. These archetypes are what are ultimately real, variation simply being an illusion that hides them. In contrast, the populationist takes the opposite approach. They do make statistical generalizations, but their results, means, and what have you are taken only as abstractions. It's the variations themselves that are real, not some completely average ideal sitting perfectly center on the bell curve. The importance of population thinking for DeLanda lies in the fact that it dispels one of the two most undesirable features of essentialism, the notion of fixed types. The other undesirable feature, the role of ideal norms, is in turn eliminated by one of Darwin's own concepts, that of the norm of reaction. The key takeaway from this notion is that two populations, when separated from one another and experiencing different material conditions, diverge. Here, DeLanda himself offers an example.
Depending on the rate of availability of a particular resource, the rates of growth of the organisms in the two communities may be different, with one consisting of smaller organisms than the other.
It's pointless to take one population as having a more ideal phenotype than the other, in other words, as having a higher degree of perfection when compared against an essence. Instead, relations between rates of change take the foreground, like rates of a certain resource's availability coupled to rates of growth. In 1,000 Plateaus, Deleuze and Guattari discuss Darwinism in detail, particularly in their Geology of Morals plateau. They credit it with dealing a double blow to essentialism, stating,
Darwinism's contributions move in the direction of a science of multiplicities, the substitution of populations for types, and the substitution of rates or differential relations for degrees.
To further flesh this out, DeLanda turns now to what are termed demes, kind of halfway individuals made up of distinct reproductive communities inhabiting a certain ecosystem. The intensive properties that compose demes, like population density, are expressed in terms of rates, such as that of growth. This rate, in turn, is defined by rates of birth, death, and migration. A deme is, in a matter of speaking, essentially a dynamic system. As such, just like we discussed in the first episode of the series, they can be considered in terms of singularities and bifurcations, the virtual long-term tendencies of a system and the changes in those tendencies under certain conditions respectively. To see how they connect, take carrying capacity as a point attractor, the type of singularity that organizes things around, well, a single point. A deme system structured by such an attractor will have a relatively stable population around that capacity, although it can never perfectly match it on account of its virtuality, discussed in the last video. Periodic attractors, described as tendencies organized around two or more states, enter the equation when
There are always nonlinear delays between the moment of birth and the moment of sexual maturity.
these delays lead to oscillations in the population of the deme system, where there isn't just one stable state, but rather multiple that it cyclically passes through. At this point, it might seem that we've drifted off, of course, from the book's central focus, multiplicities. However, there's a method to DeLanda's madness. Demes are an excellent example of how intensive processes, rates of growth, density, death, etc., find their counterparts in the virtual, like is represented by singularities. To borrow directly from the book,
Multiplicities consist of a structure defined by differential relations and by the singularities which characterize its unfolding levels.
however, there's always a gap between these virtual elements and the intensive processes that embody them. For example, singularities, by their very nature, can never be reached by the trajectories they organize. Populations in the deme system can never perfectly match carrying capacity. In a more general fashion, Deleuze also argues that intensive processes don't resemble the virtual multiplicity they embody, on account of how divergent they can be. The same multiplicity can be embodied by a myriad of diverse processes, logically leading to a myriad of different actualizations, as seen in this diagram. Simplifying quite a bit, these components that make up Deleuze's tripartite ontological schema can thus be described as a replacement for essences, the diverse processes of individuation that specify those replacements, and the individuals that are produced from their operation, the virtual, the intensive, and the actual, respectively. However, so far we've only looked at extensive properties produced by intensive processes, like population count. We haven't really made any mention of qualities, even though, as DeLanda says,
A species possesses both an extensive aspect defining its distribution in space as well as a qualitative aspect defined by population level qualities, such as playing a particular role in the food chain.
Any explanation of intensive individuation or differentiation, with a C, necessarily has to consider this qualitative side of a process. To do this, DeLanda returns to the example of embryogenesis, with a focus on two main processes, spatial structuration and qualitative differentiation. Considering the first, eggs in the beginning can be considered as forming a topological space, that is to say, a space without much structure. The things that make them up are fuzzy and ill-defined. However, as time goes on, they become more and more specified, acquiring a metric, in other words measurable, anatomical structure. Behind this transformation lie intensive processes, like those dealing with the birth and death rates of cells. In and of themselves, these processes are non-metric. They are populational and differential, mapping rates, not exact values. However, as more finished structures are formed, these give rise to more rigid, less flexible metric relations. The end product is, to borrow directly from DeLanda, a spatial structure adapted to specific functions. For example, one that acts as a load-bearing structure under gravity, not unlike a bridge or a building in architecture. It's here that qualitative differentiation comes into play. Bridges can't withstand outside forces based on structure alone. Rather, the qualities of a material that make them up matter too. Likewise, the qualities that make up the architecture of organisms are incredibly important, from those that allow muscles to bear loads in tension to those that allow bones to bear them in compression. The intensive processes that give rise to these qualities are another example of progressive differentiation.
One which starts with the population of relatively undifferentiated cells and yields a structure characterized by qualitatively distinct cell types.
In the beginning, as you might have learned in science class, cells are pluripotent. In other words, they can become any other type of cell needed by the organism they make up. For example, stem cells can be differentiated into blood cells, brain cells, hard muscle cells, and so on and so on. One of the forces behind this differentiation is called induction, which involves collectors of pluripotent cells, termed the responding tissue, coming into contact with other groups of cells, the inducing tissue. Such a process is what is shown in this diagram. Induction at its core functions through the exchange of chemical signals. In the case of blood cells, this is made through the hormone erythropoietin, also termed EPO. It's here that DeLanda introduces the biologist Stuart Kauffman's model for explaining inductive signals. Essentially, Kauffman discovered something quite interesting. Many genes act in the same recurring pattern, doing the same thing over and over again. As such, it soon became apparent that the networks they make up are dynamic, organized around a set of singularities or attractors. From this fact, it's possible to model how cells become specialized through the construction of a state space. For example, how a stem cell differentiates itself depends on the number of attractors that surrounds it in the state space made up by the network of genes that compose it. In other words, on the basins of attraction it falls into. When using this perspective, inductive signals are first and foremost triggers. They don't directly cause differentiation, but rather make cells switch from one basin of attraction to another. This is what is represented in this diagram. EPO triggering the stem cell to leave a basin of attraction that will give rise to a brain cell, in favor of the one connected to blood. In both this example and the example of a spatial structuration of an egg, we can see how the intensive, embodying the virtual, gives rise to both qualities and extensive quantities. However, this view is still only partial. This is because, outside the presence of singularities, topology has mostly been ignored, even in spite of the fact that the virtual is almost entirely topological in nature. To get rid of this blind spot, we have to turn our attention to embryogenesis once again, something that involves
looking at a developing embryo as a process of assembly of organisms, a process which must yield individuals with the capacity to evolve.
To do this, it's worthwhile to look first at industrial assembly. Here, all the components that pass along the line are firmly metric, with defined sizes, shapes, and positions. This starkly contrasts biological assembly. The components are instead defined by their topological connectivity. In other words, by their ability to be connected together, not by their specific properties. As DeLanda says, it doesn't really matter how long a given muscle is, only that it could be attached to other parts. The industrial assembly line is rigid, each component of a product moving in a specific direction being attached to each other in a specific order. In the biological assembly line, on the other hand, the situation is completely different. To use the opening passage from Difference and Repetition, in a way quite distinct from its original intention,
there is the once more calm surface, upon which floats unconnected determinators like scattered members, a head without a neck, an arm without a shoulder, eyes without brows.
Biological assembly is organized like this surface, where components randomly drift into each other and attach based on matching patterns rather than exact positioning. This is what allows for mutation and thus evolution, topological connectivity allowing a diverse range of possibilities. Possibilities that wouldn't be available if eggs were developed on an industrial assembly line. Adding on, the topological aspects of organisms never disappear. Mutation in biological assembly is always occurring, at least on the cellular level. In this way, it's impossible for something to be fully metric. There is always something unmeasurable, something non-geometric implicated in the organism. Now, we get to the end of what we'll be looking at today, DeLanda summarizing how embryogenesis models the ways in which the virtual is actualized in space. To begin with, by analyzing structuration, we saw how intensive processes are non-metric in complex and subtle ways, connecting the topological and geometric together. Next, we saw how the products of these processes are, likewise, never completely metric in nature. There's always something like mutation stopping them from becoming fully inert. Lastly, in DeLanda's words,
a finished product is characterized by qualities which also result from intensities but which are metrically indivisible like intensities.
It's in this way that the diagram we started with at the beginning of this video is only a metaphor. The movement from continuous topological space to discontinuous spatial structures, like those found in Euclidean geometry, is not as straightforward as it seems. With this, we're done with the first half of Chapter 2 from Manuel DeLanda's book, Intensive Science and Virtual Philosophy. I truly hope you enjoyed or learned something, and if you feel I got anything wrong or wasn't as clear as I could have been, please do feel free to let me know in the comments so I can do better next time. In the next video, we'll finish the chapter and pick up where we left off here, further unraveling DeLanda's mathematical metaphors. Until then, bye!